All right, we did it. Welcome to your fourth and final lecture in our Chapter 10 unit, Genetics and Evolution. Today we are going to focus on gene pools and speciation and get into how you create new species and what the purpose of gene pools and why we care about them are. So let's just get started. So first we need to get some definitions out of the way. So for species, that's just individuals that have the potential to reproduce and produce fertile offspring. That's the key point there, fertile offspring. They need to be able to produce offspring that can also reproduce. Speciation is this idea of forming a new species by splitting an existing population into something new. So this can happen in two different ways. Post-zygotic, right after birth, where two organisms attempt to have offspring but fail. That could be caused by infertility. Or pre-zygotic, which is factors that isolate the organism from mating to begin with, like geographical barriers. So a gene pool, another definition. A gene pool is all the alleles for every gene of a population that are together without being assigned to specific individuals. So these are all the options that exist for a specific species, right? So for humans, in the gene pool, think about eye color, right? There's tons of options in the pool, but you don't get assigned every single eye color, right? You get assigned one. So it's just the idea of a gene pool is everything that's possible to you, but not necessarily what's assigned to you. So individuals that reproduce contribute to the gene pool of the next generation, right? You have to have sex to contribute to the gene pool. That's just how genetics works. So why do we look at gene pools? So alleles have different frequencies. So, right, some alleles show up way more frequently in a population than others. So it shows the potential traits in future generations. You can also track the frequency of alleles over time, which shows patterns of evolution, and gene pools define species. So if organisms are reproductively isolated in specific gene pools from other species, then we can define them as a separate species. So gene pools are super important, and we care a lot about them. So I'm talking about gene pools, but like, the human gene pool maybe hasn't changed that much to your thinking, but oh yeah, stable gene pools can change over time. And there's a lot of ways that they can do it. So gene flow is when new alleles enter a population. This could be due to immigration or emigration. So this is when maybe you're introducing new animals to continents and they can interbreed with other animals. So like you bring a different variety of horse and you bring it over from somewhere and they can interbreed with a horse that we have in America, and you get new genes added to the gene pool. Natural selection is when only the fittest traits are passed on. So only the traits that are benefiting in some way. And then the smaller the population, the smaller the gene pool. And if you have a smaller gene pool, that means you have smaller variation. But that means faster change over time. Because natural disaster can just like wipe you out, a change in habitat can wipe you out, introduction of an invasive species can wipe you out, right? So because there's smaller variation, these things like a natural disaster or changes in habitats can force you to adapt and change faster because you have to, because there's just not another option. Now, if you thought you were going to get off easy in this PowerPoint, then you were sadly mistaken. So we are going to learn a new equation. So even though we've been doing like recombination and chi-squared and all of this, we're introducing Hardy-Weinberg this time. So Hardy-Weinberg is how you calculate frequencies of alleles, genotypes, or phenotypes within a population. So if the alleles of a gene pool with two alleles are given, we use the variables P and Q. And then you can figure out the genotype frequencies by using the formula P squared plus 2PQ plus Q squared. And all of that should equal 1. Okay, so far so good. So let's do kind of a practice here. If an estimated 10% of people are left-handed, that's a phenotypic frequency of 0.1. So now we want to figure out what the dominant allele frequency is. They are homozygous, right? Left-handed people are homozygous recessive for this trait. So what that means is Q squared, right? This number in our formula, Q squared, is 0.1. So you have to take the square root of that, which is about 0.32. So now I have Q by itself. Then I can do some math, right? Bring Q to the other side. So 1 minus Q, or 1 minus 0.32, will give me P. So P equals 0.68. Now, that means 68% of the people carry the dominant allele. Now you could take that one step further and really figure out how many are homozygous dominant and how many are heterozygous, but for the purposes of this question, I just wanted to know the dominant allele frequency, which would be 68%. When we do this in class, you know I'm going to get more specific than that. Now let's talk about reproductive isolation. There's two types, allopatric and sympatric. Allopatric is when a population is separated by land, so geographical, right? So this is when like land or water is going to prevent males and females from finding each other. And then sympatric is when the population is in the same area, but there's other issues playing a role. So like temporal, which is incompatible time frames. So this could mean like plants reach maturity at different times, so they're never going to reproduce with each other because they're just not in the same maturity level at the same time, so it just doesn't work. Or behavioral. So this could be like birds or something, like a bird has a mating dance, and so the male bird does this like crazy elaborate mating dance, and the female is like, what is that? That's disgusting, like I don't want to be a part of this. And so the behavior of that male bird has prevented reproduction from happening. And then there's mechanical. The bodies just can't reproduce. This could be if someone is infertile, or like you have a mule, which is never going to be able to give a viable offspring. And then there's three types of selection. So selection is talking about favoring phenotypes. So there's disruptive, and that's when two extreme phenotypes are favored rather than the intermediate middle. And so what these pictures are is the red is like your general bell curve, and the blue is what is actually happening. So disruptive is when your extremes are favored. Stabilizing is when you get even more of a favor, favoring the intermediate over both extremes. So it's a little bit more of an extreme bell curve. And then directional is when one phenotype is favored over another. So like maybe before it was favoring I don't know, the left, and then over time, it's now favoring the right. So we're kind of switching gears again, kind of reiterating polyploidy here. So polyploidy is when you have more than two sets, full sets of chromosomes in cells. So as a reminder, polyploidy is very common in plants. And because of polyploidy, you can get bigger fruit, more resistance to disease, but having these different amounts of chromosomes leads to evolution and new species being reproductively isolated, because eventually a plant that has, 8 sets of chromosomes is not going to be able to reproduce with a diploid plant. They're just too different at that point. So some advantages and disadvantages. So advantages, of course, is it forces heterozygosity. So this is hybrid vigor. This just means that you get more traits that, by being heterozygous, it's less likely that you're going to be affected by any sort of homozygous recessive disorder. So that's nice. Again, 2 kind of deals with that, prevents the accumulation of recessive mutations. And then 3, polyploidy organisms are well adapted to their environment and resistant to diseases. All huge advantages. Disadvantages, though, is your genome, like your relationship of your cell and size of genome is off because your genome is just getting massive, but your cells aren't really getting any bigger. This can change your gene expression, which doesn't always lead to good things, and your mitosis and meiosis, like your normal mitosis-meiosis events, can be disrupted because you just have so many copies of this genome. And then finally, your types of speciation. So there's thought to be two types, gradualism and punctuated equilibrium. Gradualism is thought to be the speciation that is actually occurring, but we talk about both of them still. So gradualism is small, continuous, slow changes that you don't really see, but over time you can see how this speciation has taken place. Whereas punctuated equilibrium is really quick changes followed by long periods of no change. So you see something happening right away, but then you don't see anything for a long period of time, and then you see another speciation event, and then you don't see anything for a long period of time. Now, of course, we'll be studying this for years and years and years and years, so although right now we think that the actual type of speciation that is true is gradualism, who knows what's going to happen in the future? Maybe there's a new type of speciation that we haven't even discovered yet. We'll see. I won't be around for it, but it'll be interesting to see how things change over time. Okay, that was the last of our Unit 10 lecture notes, so I will see you in class.